Hi, my name is Sarita, lecturer in Botany, Government Degree College for Women, Nalgonda. Today, in this video, I am going to talk about finance reproduction. In that, going to discuss about structure of the male cone. This finance belongs to division Gymnosperms, class Coniferapsida, order Coniferales, family Finaceae, genus Pinus. This Pinus plant body is sporophytic plant body. Pinus is a monoecious plant, that means male and female flowers are present in the form of cones or stomules on the separate branches of the same plant. Many male cones are present together in the form of clusters, each of which consists of many microsporophylls. These are the male cones. Many male cones are present together in the form of clusters, each of which consists of many microsporophylls. The female cones consist of megasporophylls. The male cones on the plant develop much earlier than the female cones. Male cones are produced in March in hill areas and in January in the plains. In Pinus, each male cone is oval in shape and not very large in size. Each male cone measured 2 to 4 centimeters in length. The male cones develop in clusters in the axils of the scaly leaves on a long shoot. On long shoot, they replaces the dwarf shoots of the long shoot, and the male cones are shortly stalked. A male cone consists of a large number of microsporophylls which are arranged in spirally on the cone axis. The male cones develop in clusters in the axis of the scaly leaves and long shoot. They replaces the dwarf shoots of the long shoot, and the male cones are shortly stalked. A male cone consists of a large number of microsporophylls. These are the microsporophylls. These are the microsporophylls. A male cone consists of a large number of microsporophylls arranged in a spirally on the cone axis. This is the cone axis. Each longitudinal section of the male cone shows central axis. This is the axis part. This axis bearing a number of small membranous, scaly and shortly stalked brown colored spirally arranged and closely fitting microsporophylls. These are the microsporophylls. This is the central axis of the male cone. This is the longitudinal section of the male cone. Two Microsporangia or pollen sacs are present abaxial surface or on the lower side of the Microsporophylls. These are the Microsporangia. Two Microsporangia are present, or pollen sacs are present abaxial surface or on the lower side of the Microsporophylls. The lower side of the Microsporophylls. Two pouch-like Microsporangia, which is also called as a pollen sacs, are present on the abaxial of under surface of each microsporophyll. Each microsporangium has many microspores. This microsporangium having many microspores or pollen grains. A microsporophyll is comparable with the stamen of the flower of angiosperms. This is the stamen of the angiosperm flowers. This is the filament part. This one is anther part. A microsporophyll is comparable with the stamens of the flower of angiosperms because it consists of a stalk. Stalk equal to this filament. Here, this stalk equal to filament with a terminal leaf expansion that is equal to this anther, the tip of which is projected upwards and called apophysis. A microsporophyll is comparable with the stamens of the flower of angiosperms because it consists of a stalk which is equal to filament with a terminal leaf expansion that is equal to anther, the tip of which is projected upwards and called apophysis. Next, the expanded end part of the, this is the expanded end part of the this expanded end part of the microsporophyll is called apophysis. This is the apophysis. It is a sterile part. It turns upward to cover the microsporangia of the other sporophyll. In the male cone, the microsporophylls are horizontally arranged on the axis. Generally, sterile microsporophylls are present at the base of the male cone. Fertile sporophylls are present at the apex. Next, move to structure of Microsporangia. This Microsporangia is a sac-like structure without stalk. It present on the lower surface of this microsporophyll. Each microsporangia consists of 2 to 3 cell thick wall with the outer layer acting as epidermis, the innermost layer acting as tapetum. Inside this microsporangia, numerous microspores or pollen grains are present. At maturity, Microsporangia split longitudinally and releases microspores. Coming to the development of Microsporangia. Development of Microsporangia is eusporangiate type. Eusporangiate method means if the sporangia develops from a group of initials, that method is called eusporangiate method. Eusporangiate method means if the sporangia develop from a group of initials, that method is called eusporangiate method. Here we can see eusporangiate type. Microsporangia arises from a group of cells on the lower surface of the microsporophyll. This group of sporangial initial takes periclinal divisions. This group of microsporangial initial cells takes periclinal division. Periclinal divisions means in the group of cell or tissue contains upper surface and lower surface. In this, periclinal division takes place parallel to the upper surface and lower surface. It is called periclinal division. Periclinal division means in the group of cells or tissue contains upper surface and lower surface and upper surface and inner surface. In this, periclinal division takes place parallel to the upper and lower surface is known as a periclinal division. This group of sporangial initial cells takes periclinal division and forms outer side parietal cells and inner side archisporial cells. This outer parietal cells takes further periclinal divisions and forms 4-layered sporangial wall. Outer layer act as a epidermis, outer layer act as a epidermis and innermost layers act as a tapetum. Next, move to archisporial cell. This archisporial initial divides to form sporogenous cells and later spore mother cell. It forms spore mother cells. The spore mother cells takes meiotic division and forms pollen grains which are known as a microspores, which are yellow in color. Microsporangia ruptures and microspores are released. The dense pollen grains released are referred to as yellow clouds or shower of sulfur. Pollen grains have wings that are useful to dispersal. These pollen grains have wing-like structures that are useful for dispersal. This is about these are the pollen. These are the pollen grains. Each microspore or pollen grain is a rounded and yellow color light uninucleate structure with two outer coverages. That is, outer layer is called exine and innermost layer is called intine. Each pollen grain has two wings. Each pollen grain has two wings, two balloon-shaped wings like airbags. These wings help in floating and dispersal of pollen grains. This is about structure of the male cone. Finus, structure of male cone structure. Thank you.